Hello everyone. Welcome to Scholar Labs, a unique virtual platform that brings science experiments to life through immersive 3D augmented reality and engaging content. Our simulations ignite curiosity and enhance classroom experiences, crafted by expert teachers and accessible on all devices without special equipment. Today, I will be taking you through the simulation Asexual Reproduction under the topic Reproduction in Animals. In this simulation, you will be able to understand various modes of asexual reproduction in animals. Before we run the simulation, let me take you through some important control buttons. By clicking on this I button on the top right corner of the screen, you can view the objectives of the simulation. You will also get step-by-step instructions on how to use the simulation. The buttons on the right side of the screen can be used to view the simulation from different angles. You can use the mouse scroll wheel to zoom in or zoom out. Else, you can also use this zoom slider on the bottom left corner of the screen to do so. So let's begin. Reproduction is a process by which living organisms produce new individuals of their own kind and maintain their existence generation after generation. There are two main methods of reproduction in living organisms. The first one is sexual reproduction. It is a type of reproduction in which two different sexes are involved, that is, male and a female. It involves the fusion of gametes from two different parents and results in the formation of a new organism which is genetically different from the parent. This method of reproduction will be discussed in detail in another simulation. Besides sexual reproduction, the other major type of reproduction seen in the animals is asexual reproduction, which will be discussed in this simulation. This type of reproduction is mostly observed in lower organisms and unicellular microbes. In this process, a new individual is formed by the involvement of a single parent. The individuals thus produced are genetically and morphologically similar. The cells divide by mitotic divisions and no fertilization takes place. Here we are going to discuss three types of asexual reproduction, that is binary fission, budding, and cloning. Binary fission is a mode of reproduction in which an organism reproduces by dividing into two individuals. Click on this slide to place it under the microscope and then click on this eye button on the bottom of the screen to see how binary fission takes place in paramecium. You can see a mature paramecium here. Binary fission is the commonest type of asexual reproduction in paramecium. It occurs during the favorable conditions when food is available in large quantities and the temperature is also favorable. Click on this button to observe the various stages of binary fission in paramecium. In this process, the macronucleus divides transversely, amitotically, by simply becoming elongated and constricted in the middle. In the next step, oral grooves begin to form. Then, nucleus divides fully and constriction furrow deepens. Ultimately, the cytoplasm is completely divided, resulting in two daughter paramecia of equal size. These grow to full size and again divide by fission. Click on this button to get back to the first screen. Click on this menu button on the top left corner of the screen to explore the other methods of asexual reproduction in animals. In budding, the offspring grows out of the body of the parent. It remains attached to the parent until it matures. After maturation, it detaches itself from the parent and lives as an individual organism. This form of reproduction is most common in hydras. Click on this slide to place it under the microscope. Now observe the budding process by clicking on this eye button. A tiny bump appears on the body of the hydra known as bud. It then grows in size and develops tentacles. It then detaches itself from the parent hydra to become an independent individual. Let's go back to the main screen. Another type of asexual reproduction is cloning. It is the production of an exact copy of a cell, any other living part, or a complete organism. Cloning of an animal was successfully performed for the first time by Ian Wilmot and his colleagues at the Rosalind Institute in Edinburgh, Scotland. They successfully cloned a sheep named Dolly. Dolly was born on 5th July 1996 and was the first mammal to be cloned. During the process of cloning, a cell was collected from the mammary gland of a female fin Dorset sheep, which has been represented here as sheep A. Simultaneously, an egg was obtained from a Scottish blackface ewe, that is sheep B. The nucleus of this egg was removed or enucleated to make sure that none of sheep B's DNA could be passed on to the next generation. In the next step, the nucleus of the mammary gland cell, which was obtained from sheep A, was inserted into the egg of sheep B, whose nucleus had been removed using an electric shock. The chimeric cell thus formed started to multiply. The cluster of cells produced were then implanted into the surrogate sheep, that is sheep C. Development of this egg followed normally, and finally Dolly was born. Though Dolly was given birth by the Scottish blackface ewe, that is sheep B, it was found to be absolutely identical to sheep A, that is Finn Dorset sheep. Since the nucleus from the egg of sheep B was removed, Dolly did not show any of its characters. Dolly was a healthy clone of Finn Dorset sheep who died on 14th Feb 2003. Thanks for watching.